Welcome, and thank you for joining us for the R&D Scientific Seminar Series today. Our special guest speaker today is Professor John Wikswo. He's a university professor at Vanderbilt, and he's the founding director of VIIBRE. That's the Vanderbilt Institute for Integrative Biosystems Research and Education. That program has been tremendously successful at Vanderbilt and has trained a variety of really great engineers. Including myself, actually. I was one of John's ex-students. I like to think of myself as one of his favorites, but I don't know, maybe he thinks otherwise. He did receive his PhD from Stanford University. He's published more than 200 peer-reviewed papers. He's a fellow of 7 different professional societies. He holds 37 patents now. And he'll be telling us a little bit about his research today. He's primarily in the field of systems biology, focused on organs on a chip. John, thank you for joining us today.
Okay, Louis, you definitely are one of my favorite students, in part because you were always making your own path through science, and I just was deeply impressed by how thoughtful you were as a student in the classes you took with me. Okay, so I'm going to talk about automated microfluidic pumps and valves for interfacing bioreactors, analytical instruments. The official disclosure is I'm funded by lots of people, agencies. I have in the past been funded by others. Some of the technology you see has been licensed to Kiatech and Agilent, and others is currently under license for development as a product by CN Bioinnovations in England. These are my views and no one else's. Okay, I can't figure out exactly what anybody in the audience might want to hear, so I'm going to fly through the breadth of my work. And the idea is you'll get an overview so you can decide what'll be of interest. I'm going to go too fast, probably take notes, but I want you to be able to figure out what might be of future interest, and then Louis will set up one or more meetings to go over the details. So I'm going to weave together 7 themes. How do we do biology, organs on a chip, automated biology, fluidic control, and ending up with my current project in chemostats. So people have been studying biology with people, but it turns out from the point of view of pharma, they're not isogenic. You can't get controls. A lot of data you can't get. Animals are not people. Cells in vitro have a few problems. They have cancer, they're inbred, they're diabetic, they're potatoes on a stiff couch with little exercise. They have neither gender, sex, live in the dark. They suffocate in an increasingly acidic environment. They live in their own excrement. They never bury their dead. They talk to cells of a like mind. But you can get wonderful, reproducible, statistically significant results, and the question is, are they relevant to human biology and disease? So that list, partly in jest, is published if you ever want to use it. So looking at the existing technology, high throughput screening is phenomenally successful. This is the AZ library, and using acoustic spitters to fill the wells. The lessons you learn, and so my notation today, basically, in general, blue is good news and red is a source of concern. So HTS with robots is industry standard. It uses biology on plastic. The robots are big enough to kill you if you stand in their way. And the fundamental question is whether the idea of culture, expose, and measure is the correct way to understand some of the subtleties of biology. So realizing that I'm talking to everybody working at an absolute giant in genetics, I just wanted to point out that you folks and others are doing nanoliter, picoliter, single cell biology. It's extremely important. I think it's going to be the critical in our understanding of both obvious heterogeneous cell populations and, more important, populations that are assumed to be homogeneous. So I'm not going to say any more about these fantastic techniques, just that I respect them highly. Now, some of this nanoliter, picoliter fluidics can be done with a Quake chip, and we've built devices with Quake chips that are fairly sophisticated to formulate solutions for experiments. The only trouble is that the solenoids and tubing are bulky and get in the way and are not suitable for extending to very large populations of experiments. So the lessons I've learned from nanoliter, picoliter, and single-cell biology is that from the point of view of fluidics, pneumatic microfluidic pumps, valves, droplet technologies that control nanoliter volumes are very good, but they do not scale to the volumes of hundreds of microliters that are typically used in today's well plates, the larger well plates, and particularly for experiments where you're interested in doing untargeted transcriptomics, proteomics, and metabolomics. And that's what I've been worrying about for a bit. So the organs on a chip business has been running for 15 years. The past 10 have been very intense. Fundamentally in biology, you have a rule of thumb that I was taught years ago, is that a cell has a volume of a picoliter, and it requires a nanoliter of media each day, both to get sufficient nutrients and to have a place to pee into that you don't poison, the cell doesn't poison itself. So the way this is typically done in biology is there's a, let me go find my, you have a layer of cells down here in the bottom, and you then put 1,000 times the media on top of it. And the only trouble is, well, the same rule happens if you take a 5 nanoliter spheroid and drop it into 5 microliters. This allows you to do 1 or 2 days between fluid changes or between to the end of the experiment. The trouble is that anything the cells secrete are diluted by a factor of 1,000, and you have time-dependent concentrations of everything. So the argument on microfluidics is that if you bring the lid down, what you end up doing is you can have a much lower cell to media ratio, but this original rule still applies, which means you now have to do 5,000 fluid changes a day. And that's more than you can ask most graduate students, definitely more than you'd ever ask Lewis, because he'd ask why and come up with a different way. But the low dilution has certain merits. So getting away from biology on plastic, 3D organoids are becoming extraordinarily important. These are images from an experiment done by colleagues of mine at Vanderbilt, where they're growing mammospheres and looking at dose response curves to an anti-cancer drug. If you want to get a picture on the cover of Nature, get a beautiful brain organoid. They have lots of advantages and they have a number of disadvantages. And in my point of view, it's hard to do barrier functions and you can't apply mechanical stresses and you can't couple compartments. If in fact you're trying to do a 1536 well drug screen, this is probably exactly what you want. But if you want to do more, if you need more sample mass for quantitative analysis of one form or another, you need to think about organ chips. So my goal over the past 17 years, 7 years, has been to, or actually 10 years, has been to launch 2 new species, Homo minutus, the microhuman, and Homo chipiens, the nanohuman. So here's a human holding a millihuman, holding a microhuman, holding a nanohuman. And each of these are called a homunculus. So organ chips come in a variety of flavors. I think everybody in the community is trying to get 2D, 2D plus, which is basically a thick layer on a barrier, or 3D, where you're trying to recapitulate the in vivo environment. And pharma sees both pharma and toxicologists and scientists see the benefit of getting higher throughput, lower course, deeper analytics, and pharmacokinetic and circadian control. So this is the future of what we need. Now, looking at the organ chips, they have their own sets of advantages. They're better than 2D biology. You can use minimal media volumes. They are being vascularized by other groups. You could build an animal on a chip. They require microfluidics control of one form or the other. They're not high throughput. Today, it's hard to get the cells out. These assays are about as expensive as an animal study, and there's no way to gene co-expression network analysis validation yet. and you can't transplant an organ on a chip into a person where you could seed organoids. So I spent some time working on trying to understand how big each organ should be, and you do the allometric scaling laws. But the insightful observation that came out in a paper at the same time was from Shu Takayama, now at Georgia Tech, where he looked at the basal metabolic rate of an organism versus the mass of the organism, and you get a power law of mass to the 3/4. What that means, though, is if you now look at the organism mass versus the cellular basal metabolic rate, just divide by the number of cells. In vitro, everybody runs with a flat metabolism, whether you're looking from a paramecium or an elephant, where this is actual biology. So in vivo, cells have limited resources. In vitro, everything is running resource-rich, typically with high levels of glucose and oxygen, and cells behave differently. So there is a whole zoo of organs on chips, lung, coming out of Emulate various organs, coupled organ systems from Asperos, Tissues in Germany, Nordis in Seattle, in vivo coming from Huntsville, Mimitas from Netherlands, CN Bio from London. So there's some challenges. I'm not going to go into them at length, but quickly fly through them. Size, fluid control, analytical chemistry in small volumes, the blood surrogate for all the organs, putting them together, controlling them. What do you do for the organs you don't have? How do you couple them? How do you diagnose them? How much will it cost? And most important, how accurate a model can you produce or do you want to produce? So that was in 2000, that's 2013. A few years later, I revisited it, and a lot of the previous problems are being addressed. You still have issues on the cells and tissues, and I think largely the hardware issues are very much in need of address. And today, the ones that I'm looking at is, okay, you want to look at COVID or SARS-CoV-2, you got to get your models into BSL-3. You got to make the pumps and valves both controllable and low cost. You got to get rid of PDMS. You have massive amounts of data. You got to find the right cells. This is a biggie to get the adaptive immune system. How do you control it? We'll talk about the neurohumoral connections, the need to be able to run 10,000 models, and closing the hermeneutic circle of biology. So in 2017, Dave Watson, then at Lilly, Rosemary Hunsiger at NIH, wrote a review that reversed the order of importance. The organs on a chip programs were established for toxicology, but in fact, as we got to know what they could do, they focus now on disease biology, pharmacology, ADME, PChem, and toxicology is coming in third, a strong third, but it's still a third for a number of reasons, including inertia in the industry. Now, if you can get the organs on chips to work, they may reduce costs through drug toxicity and efficacy improvements. You're going to be able to measure tens or even hundreds to thousands of cellular variables, and this is where we're heading towards the kinds of things that Illumina does so well with single cell, massively parallel RNA-seq. And what you're trying to do is understand biology, get drug effects, get mechanisms of action, understand unknown effects of environmental toxins, and get new models for disease. This is a point that needs to be addressed, is does your system replicate in vitro data from primary cells and cell lines? Maybe they shouldn't. Do they replicate in vitro animal data? Ideally, if you have animal chips. And do they mimic, most importantly, human clinical data? And then again, there's the weighted gene co-expression network analysis. Questions, does human on a chip equal human? And an important question that would allow transition of information in pharma scale-up. Can you show that mouse on a chip equals mouse? And then you can hope that human on a chip equals human. So there are problems on organs on a chip that are important with respect to organoids, capabilities, not problems. If you want barriers for shear flow, if you want a mechanically active system, if you want to have things that are coupled together or complex topologies, electrical recordings, or coupling the organs. Those are just things to remember. Now, if you look at what's at the cutting edge of microphysiological systems, the two that I think are the hardest is the full metastatic cascade, where this is where people are working heading in this direction. The fundamental problem is a lot of people would like to look at immuno-oncology drugs, but nobody has yet figured out how to build an innate and adaptive immune system, particularly adaptive to coupled organs on chip. There are moves afoot to do donor-matched leukocytes to iPSC cells used to create the organs, which is a good step in that direction. So the take-home lesson that I want everyone to get from today is that I'm going to try to show you that we can build multi-channel fluidic pumps. We can build multi-channel microfluidic valves. We know how to do digital control. And the goal is to take, instead of taking a well plate to a single large robot, make it that you bring the smaller robot to the well plate at a cost that allows you to go massively parallel and independent experiments. Going back to the volume problem, if you're doing pipetting between reservoirs or pressurized reservoirs, you have to be careful because you still have large volumes of inlet and outlet media. So while the dilution is fine here, it's not here. The direction people are heading is to couple organs on chips with small volumes of media, as small a volume as possible. But given the nanoliter picoliter rule, you still have to deliver fresh media and give your coupled organ on a chip a place to pee into. And it's more serious because organ on a chip livers are not going to do as much metabolic cleanup, and the kidneys will not be doing selective removal of waste products. But this is where the industry is heading. Now, in terms of the volume problems, conventional cell culture, these are the rules, 24 to 40 hour media changes. It turns out people who do the video intra-incubator incucyte microscopy point out that the greatest trauma to a cell is to feed it every day. And you have these large dilution effects. Perfused MPS cell culture is going to let you get transient signals cleaner. If you have recirculation with replenishment, you'll create steady state conditioned media. But you may want to wash things away faster than diffusion, and you also want to be careful that you don't wash things away that you didn't want to wash away. So given this picture, how do you keep the single and coupled organs alive? Simply, you have an arterial reservoir, an organ, and a venous reservoir. If you have an organ that has a barrier, you have two reservoirs at each end. And the obvious way that has been done for years that I still continue to do in our lab is syringe pumps. They're the fastest way to get into the business. They have a large number of disadvantages, including the shock of having to change syringes and making it hard to recirculate. Luke Lee's group at Berkeley spun off a company called CellAsic that got bought by Melekpor, that got bought by EMD, that takes a specialized well plate with pressurized reservoirs, a pile of tubing to run 4 parallel experiments. And Nordis took the idea of the pressurized reservoir and has built devices that use pressurized reservoirs and fluidics to run organs on a chip. Do you need recirculation? So recirculation in theory is simple, but you do have to include a bubble and debris trap. And when you get to this point, not only do you have to do recirculation, you have to remember you have to put in fresh media and give the place a place to pee into. So there are advantages of recirculating and there are disadvantages. And the trick is to figure out what's best for your problem. Now, multiple organs. Mike Schuler at Cornell has been pushing multiple organs since the very beginning, 15 years ago. And this is one of the more modern advanced chips that he and Jay Hickman are building at Hesperos. Mimitos builds very simple organ on chips based on a well plate technology. Both of them use shakers to tilt and get media to flow from one to the other. A group in Germany uses peristaltic pumps and something that's functionally equivalent to transwell inserts to have a blood circulation and a urine circulation. CN Bio, coming off of the MIT DARPA project that was parallel to the Harvard effort, has built what's becoming a very general purpose platform that allows localized recirculation in well plates and connections between devices. So the effluent from here goes to there. The advantage of this technology is it gives you enough cells that you can do analytical chemistry on it, and more important, you can grow the organs up independently and then put them together when each organ is right. The Harvard group spent a great deal of effort trying to figure out how to pipette media from one to the other, and built a robot that fills an incubator. But there are disadvantages in any pipetting technology. Colleague of mine who built the high throughput screening core at Vanderbilt pointed out he learned he was doing affinity chromatography in his pipettes. And you have to have reservoirs that are open with evaporation, or you got to have a septa, and you might have to do thousands of penetrations. So I think pipetting is not the way to do it. The key thing is that once one way or the other you build a multi-channel system, you can start doing pharmacokinetics for the interactions. And some of the prettiest work is coming out of the Laufenberger and Griffin groups at MIT. Now, let's see what happens when you try to put it all together and you look at the microbiome, gut, liver, immune, brain axis. So why the microbiome? There's a rapidly growing evidence of microbiome's influence in behavior, mood, decision-making, CNS diseases, big-time CNS diseases, and a whole pile of non-CNS diseases. From the point of view of FDA, there are 21,000 human genes, 4,500 of which have been considered druggable. There are 700 proteins that are currently targeted by FDA-approved drugs. Yeah, at a first guess, you would argue that there are 3 million genes in a microbiome, but if you look at small proteins that are regulatory and are coded in the genes, you can argue between there are 20 or 40 more million genes available for targets, which is a growth industry for people who happen to be sequencing bugs. Now, if you look at the microbiome gut, liver, brain axis, this is the central dogma for a human: genome to transcriptome to proteome to metabolome. And you have in parallel a microbiome for all the microbiota that are in the human. The key thing is that the metabolites from the human microbiome feed the gut, provide nutrients to the gut, and the metabolome from the gut serves as nutrients to the human. Everything is coupled together with the environment, and it's all tied together in a tight loop. So it's a marvelous place to go gene hunting. Now, suppose you wanted to recreate that, and what you'd end up doing, you have to worry about the enteric nervous system, which has more neurons than the spinal cord, and the autonomic system that connects it to the brain. This is a very hard problem, especially when you're adding a microbiome here. So if you want to look at a simple problem where you are basically carrying your media in little jugs from one location to the other to a mass spectrometer, which I call functional coupling. So this is the functional coupling from the liver to the brain, the functional coupling from the gut to the liver. That experiment has been done in a collaboration between Vanderbilt, Hopkins, Baylor, Pittsburgh, and Washington. And we had basically 4 different bioreactors: the gut, the liver, the kidney, and the brain, all running in different geometries, with the gut running in a transwell, kidney running in a Nordis chip, liver running in a different flavor of Nordis chip, and our blood-brain barrier. So the idea was, in this workflow, you would take a drug, you give it to the intestine, you would take the media from the intestine and give it to the liver. You would then take the liver media and give it to the kidney and the brain, and then you would mass spec the daylights out of it all. And we did 3 experiments: vitamin D3 transport, terfenadine, and trimethylamine. This basically said you put this into the gut and it shows up in the kidney and the brain where you expect it. You put terfenadine, which is Seldane, which led to the Seldane cardiac arrhythmia and black box. gets converted into fexofenadine, and fexofenadine does not cross into the blood-brain barrier. It's eliminated by the kidney, and fexofenadine is now the replacement for terfenadine. TMA is a trimethylamine. It's the product when the gut microbiome digests carnitine and choline. So we didn't have any bugs in our setup, so we just basically fed TMA to the intestines. The intestines transported it across the barrier. The liver metabolized it into TMA and TMAO. Everybody expected that both would be eliminated by the liver, by the kidney. Nobody knew what the TMAO would do to cross the blood-brain barrier. We did the experiment and found 26% penetration of TMAO into the brain. And interesting enough, later that year, a group did 50 human CS spinal tap experiments and confirmed our prediction. So to the best of my knowledge, this is the first prediction in an organ on a chip that has been confirmed by a subsequent clinical study. Now, it's not going to be hard, given the technologies I've already shown you, to functionally couple all of these organs together, and various groups are heading in that direction. The trouble is, if you want to add the enteric nervous system and the autonomic system to get autonomic control, I know of no way to run a neuron in one organ to a liver in another organ and gut somewhere else. The question is, how do you fake it? And a colleague of mine in molecular physiology and biophysics introduced me to DREADS, designer receptors exclusively activated by designer drugs. Neither the receptor or the drug have any ligands or effects in humans. So this is chemogenetics. And the beauty of it is these groups have built DREADS that replicate G protein coupled receptors, which are the things that connect the autonomic nervous system to the gut. So you can start imagining faking the autonomic nervous system using chemogenetics. Okay, so the lessons, there are a number of lessons learned. They can be coupled. It's probably wise to grow the organs individually. I haven't talked about it, but never ever try to build an exact model of a complete human. It'll be too complicated to understand, and you'll fail trying. So if you look at these modular bioreactors, how do they help the general problem? So we've been working on 5 different areas and learning how to build better with collaborators, building better iPSCs, building better bioreactors, developing control hardware, improving analytical chemistry, and trying to translate it out into the world. So the approach that's been running now for a dozen years is make general purpose components, treat an organ as a module, couple the modules together, figure out how to do sensing. We're heading towards closed loop control. The missing organ microformulator was the thing down here that I coined using a gadget we had built for electrochemistry to essentially address the concern of any reviewer says, oh, but you don't have my favorite organ. And my response is, we'll fake it with the missing organ microformulator, and then the analytics. So the blood-brain barrier is basically a chip that has a vascular component separated from a barrier by the brain side, little bitty channels where appropriate, cells in the appropriate places, endothelial cells, astrocytes, pericytes, neurons, heading towards microglia. You can measure all sorts of stuff, the barrier tightening with time, being disrupted by glutamate, ascorbic acid, take your vitamin C. LPS from a bacterial infection would cause the blood-brain barrier to get loosened. Ascorbic acid tightens it up. You can look at penetration of terfenadine versus fexofenadine, and you can look at combined assaults of basically glutamate and acidification as a model of stroke. We're also building what we call a puck bioreactor, which is openable so that you can get into it to get the stuff out. And the advantage of this is the hardware for this costs less than $50. This is what it looks like, and you can open it up and get stuff out. So we're bringing that online. The first people to bring it online are my graduate student, Nicole Mashinsky and Melissa Farrow in the Med Center, where they're growing GI epithelial cells and looking at cells before and after flow and showing that they can extract lots of proteins and lots of metabolites out of one of these chips. And if you compared metabolites, we find more things in the metabolite from the puck bioreactor than in the tissue. So the approach is working. And there are all sorts of different flavors you can do with these pucks. A lot of the stuff I'm showing has pending patents on it. I don't have the labels on all the pictures. So basically, the lesson learned so far is that this is a whole complete different set of challenges. And from my point of view, it's okay. For some people, it makes it hard. These experiments run for days and weeks and will require all of this stuff. Not everybody realizes that they actually have to have it to scale the systems up. So to scaling up, a number of years ago, a student of mine invented a pump that was basically it's a peristaltic pump that's functionally equivalent to rolling an orange between the palms of your hands. You have a rotating disc and a peristaltic pump. It worked quite well. We started building controllers. And the breakthrough was when one of my postdocs invented what was called the normally closed rotary planar valve, which is a valve. It's more of a toggle than it stays open when you want it open. It stays closed when you want it closed. And it's actually a very good valve. We've done some very quantitative tests on it. It's got very low leakage. It can do balanced reagent delivery. This is all done with induction plasma fluorescence, and the switching time is well below 10 seconds. So once we had the pumps and the valves, when Don Ingber wanted to go after the DARPA proposal, and I volunteered to help, I realized you could build complicated systems that would be a complete organ chip with a perfusion controller that allowed you to couple organs and a microclinical analyzer to do the electrochemistry to see what's going on. And that work has now proceeded to version 2 motors and controllers, to version 3, and the most important advance from my point of view recently has been the development of spiral pumps. In order to make things easily sterilizable, we're putting all of our motors in a wipe sterilizable container. We use what's called a through-plate fluidic, which makes it very easy to insert new cartridges for pumps and valves. We've gone into production of this hardware, even though we're an academic lab. One of the troubles with peristaltic pumps, and this blue line was from the best pump we could produce up to this point. It would have a large starting transient when the balls rolled on or off of the channel. So one of my engineers suggested that we go to spirals, and the spiral pump works better than we could have ever hoped. No spikes, some oscillations. They're just extraordinarily clean. And this was, we're trying to refine it even further, but this is the channel-to-channel variation in the pumps. And we're now testing, we've demonstrated and are building better molds for a 12-channel pump. You can also make weird valves. This is a make-before-break valve if you wanted to be able to switch from one supply to the other without interrupting the flow. This is a pair of sampling valves where you go from here, where the output of every organ is going to a common drain. These are the organs or measurement channels. And you can select one to send off to another source for analysis. And here's what's called a cut-in valve. It's a similar theme that you can basically do the fluidics just by rotating this orange disc from one port to the other. And we also have ways to do it random access. In order to get cell count up, we've figured out how to build what we call a 100-channel valve. And what happens is you actually move the valve port right under the ball. So the ball shuts the port on the valve, make things really compact. We've been using these controllers now for a dozen years. We've made more than 80 of them. They had some problems in that they were just big and awkward. And we decided to move to a Mechaduino, which was a commercial motor board. Unfortunately, after a couple of years' work with the Mechaduino, and we found out that it had was missing vendor-specified blocking capacitors, and the motor driver chip was in very short order. We've been working on buttoning together all of it on our own electronics with a communications board, a controller board, actually a calculation board and a controller board. And we're currently funded by NASA to build a prototype to fly on a Blue Origin suborbital flight of a device that would have multiple bioreactors and media collection from both sides. It's all got to fit inside of a locker that would eventually be on the space station. And Ron Reiser has figured out how to make microfluidic molds that have the pins cast in place and the things fold up. So this is an early prototype showing how it folds up, and you can do some rather cute fluidics. This being 2 valves and a pump. Different views of the same thing where we have bags, a bag house and a controller house. And this is the device that we're in the process of getting ready to move to a printed circuit version of that controller and fly on Blue Origin. Other games you can play is once you have a pump and a valve, the valve serves as a check valve, and you can pressurize reservoirs and sequentially revisit them and then charge all the reservoirs up. You can build a microdialysis imager where you have a pixel of diazolate that goes in and out. This is the supply. This is the output side. This goes to the analyzer. This is your dialysis membrane. This is your layer of cells, and that's your lid. There's a trick you can play that's like a FIFO, first in, first out reservoir that pushes the samples out as you accumulate enough volume to analyze using the tubing as a collector. And it's just a matter of plumbing and electronics to make it work. I know that Illumina is interested in getting rid of PDMS or staying away from it. We know we can build our pumps from either rigid material or styrene, ethylene, butylene, styrene. We've done some prototypes on that. We can do the pucks out of a variety of materials, including machine cyclic olefin copolymer injection molded SEBS. And we're working with Tom Gaborski to build some super duper membranes out of parylene. Analytics. So these are all the techniques that people are doing for analytics. This is basically Illumina, and this is the rest of impoverished biochemistry. The sensitivity of the assays is set by the cell volume to media volume, going back to the earlier nanoliter, picoliter rule. So there's the microclinical analyzer, which was basically doing electrochemistry with calibration vials because the sensors would drift. I said, great, calibrate them. And I said, great, put the same device down here, fill this with hormones, and you've now made a missing organ microformulator. So this gadget works pretty well. This is a paper that's under review right now that shows we can build a glutamate electrode and show that glutamate metabolism is different for normal versus tuberous sclerosis iPSC-derived astrocytes. In terms of analytics, there are all sorts of things we've measured. We can look at barrier tightness, dose response curves, mechanical activity of cardiac tissue, model it even. There are all sorts of measurements you can get, and metabolic activity, morphology, and mass spec. I happen to be a strong believer in untargeted metabolomics as basically a student of John McLean, who's now a chair professor at, I've learned a lot from John, who's taught me as long as he's been at Vanderbilt. We built a gadget that would have a bioreactor with T cells in it that would pee into an automated desalter that would get injected into a Wooters ionobility mass spec, and we could pull out all sorts of stuff from the exometabolome in near real time, every 10 minutes, pull out samples. We then went a little bit further and came up with a workflow for the media out of the blood-brain barrier and showed that we could see, both with a gene ontology analysis and a network analysis, we could see the response of the blood-brain barrier to a cytokine cocktail. With a lot of funding from DARPA, we're just wrapping up a project that basically said, can you get mechanism of action instead of 10 years in 30 days? So the idea is we worked out the workflow that effectively allowed us to go with gathering up 250,000 species and coming up with different networks And this led to an expansion of the mechanism of action, a purported expansion of the mechanism of action for cisplatin. Okay, sample volume is a problem for proteomics. The McLean and Caprioli groups have gotten to the point where you can take 5 micrograms of protein from 10,000 THP1 cells and pull out 6,000 species in a unified sample preparation. So this is really a great advance. It tells you you can do untargeted metabolomics and proteomics with 10,000 cells. So the lessons learned on this is that we are building race cars. We're not doing what can be done with simple high throughput screening, which is a massive project. But Andrade says to win, you have to finish. What constitutes finishing? You have to show that your organs metabolize your drug. Its metabolites may or may not cross the blood-brain barrier. They may not affect the brain. You got to match PK. Your drug is actually doing what it needs to do. And so efficacy is a big deal. What do other organs do to a prodrug? Predict something was not previously shown in humans. We've got that one checked off. Replicating in vitro, something only seen in vivo, and keeping your cells long enough. So this is what it's going to take to finish the race. Now, going to drug pharmacokinetics. This was the missing organ microformulator. This was a calibration plot, and I showed, or Cliffle's group showed, that the gadget we built for them, you could oscillate the valve and make oscillating concentrations. So that's the widget. And suppose, though, you want to, so I mean, that's basically our analytical device and also our missing organ microformulator. So how do you control the time course of concentrations? We got the pumps and the valves. We got the controller circuitry. We got the software to drive the pumps and the valves. And that leads us to the microformulator. So Matt Wagner basically said, John, your missing organ microformulator is great. I need 96 of them. And I said, why? And he said, because I want to be able to do the PK profile. And I also want to do the optimal dosing for a drug. So we looked at it. We had the 5-channel valve and the 1-channel pump and had to build something like this. And it was going to be possible, but very, very hard. And Ron says, great, I'll build you a 25, Ron Reiser said, I'll build you a 25-port valve. And there's the, it was called the Beast by AstraZeneca, 192 tubes, 24 stepper motors. It filled an incubator shelf, and they ran it until it wore out, which was a very beautiful set of experiments. The net result of this is that we went, Wagner asked us on the 15th of May. The purchase order was on 15th of, May 2015. Purchase order in October of 15. We delivered it. We delivered that to AstraZeneca in January, and the next year we won an R&D 100 award for it. Our current ones, we have better ways of doing the plates. This is our family of microformulators. We have software that now lets you dial in a PK curve. And what can pharma do with it? This is a fascinating graph because it says basically 6 different formulations of insulin all have different PK curves. And then if with a microformulator, you can say, well, given whatever form of insulin I have, do I want to change the PK curve for better or worse? So one of the experiments was if you do drug effect on tumors, this is basically drug concentration in a well, flat because there's no liver or kidney. This is what you need from the animal. So you fake that with the microclinical analyzer, or microformulator. This was tumor growth rate versus different drugs seen in cell culture, and this is what they found in the mouse. It was a perfect match except for red, and I was told, don't worry about red, it's the drug's fault. So you can now, in vitro, you can replicate PK curves and study things that previously required animals. Another example coming out of CNBio, which is licensed technology looking at a PI3 kinase pathway. This is the literature PK curve for a PI3 kinase inhibitor. And this is what they simulated. And this is the PI3 kinase signal or concentration as a function of time after dosing. And this is what they found in vitro. So this is the already launched product from CN Bio that runs the MIT design system, and they're getting ready to launch a version of our microformulator. Circadian rhythms, the best example of circadian rhythms is the distributed circadian behavior in diabetes. A group of my students in a class several years after the one that Lewis took figured out a map for endocrine connectivity and decided you didn't need to have the entire system. You only needed to do the things that were immediately fake, the things immediately upstream of your end organs. And so here's the liver, and those are the things that you have to control to create those rhythms. There are a lot of problems in differentiation of induced pluripotent stem cells. And one of the challenges is optimizing the protocol, because it might take weeks, if not months, to differentiate some cells. So the microformulator would allow you to go in and turn all the knobs. And if this is the Waddington epigenetic surface, you're trying to steer down that groove, the underside of Waddington's surface has all these sticks. The key thing is those sticks are not orthogonal, and the net result is controlling them as a challenge. So we think we can do that with the microformulator. This is a cartoon of what we're trying to do. These are the first systems we've delivered to Ethan Lippman. However, this whole approach isn't going to work on 10,000 perfused. Everything I've said so far doesn't work on 10,000 perfused organs for a variety of reasons. So let's go look at automated biology. So in 2006, I gave myself the challenge: can I design and build a hybrid silicon biological system that proposes and generates models and conducts experiments on itself to identify the underlying equations that govern cellular dynamics? I guessed in 2006 it would take 3 to 5 years and $3 or $4 million. I'm probably off by a factor of maybe 3, but it's not impossible. The one where I made the mistake, though, was I guessed I could do the intracellular problem in 15 to 20 years with 15 to 20 million in 5 to 10 years. I think I'm off on both of those by an order of magnitude. And you can definitely see how Illumina's role in trying to understand these kinds of pictures. So this is the classic map of metabolic pathways, and I had it on my conference room table long before it got as cluttered as Lewis remembers it. And a friend of mine walked up to one of these arrows and he says, "That arrow is my career." That's the problem with these maps. So if you try to say, generate one of these maps and calculate biology, you have to put in all of these terms. And I argue that if you were going to do that foolhardy exercise, you would come up with Avogadro's number of coupled partial differential equations. Now, Ross King, now at Chalmers University, previously at Manchester, and at another British university, first developed what is known as the first truly robot scientist. It looks like a high throughput screening station. It's got pipetting arms, it's got fluid handlers, it's got different fluid handlers, it's got centrifuges, it's got refrigerators, it's got incubators. It also has some very smart software, and it basically formulated on its own 20 hypotheses and tested those hypotheses by making 6 million optical density measurements, created 26,000 growth curves, and proved that the identity of the genes that encoded 13 orphan enzymes. So this is really a marvelous machine. He then moved it to Manchester, where I got to see it, and went going for basically repurposing of drugs or understanding various drugs. And they just published an elegant paper that talks about the design build cycle of 3 different cycles to get to a improved understanding of a particular mechanism in yeast metabolism. And they did this and pointed out they had gotten 3 cycles down and only had 10 to the 4th to go. So that is really the ultimate challenge in biology, is the fact you can be very proud of 3 cycles realizing you have 10,000 to go. So the lesson learned is that to automate the inference, you have to have closed loop control of biology, which is currently very hard. Ross is doing it. Automated design of experiments is hard. Ross is doing it. All of this stuff is fairly standard. I would argue that you need new types of data from new sensing modalities. And that's where, for example, RNA-seq and the other related studies are so important. Automated data mining, and then the tissue culture. So going to closed loop control, if this is a hypothetical cell that has internal pathways, you want sensors for those pathways, you want to knock them out with a knockout or a drug. You want to have an actuator that replaces the knocked out process, and if you can loop it all together, you basically can understand what's going on if the knockout works as well as the original, because your sensor actuator integration and feedback is doing the correct things for biology. So again, from the point of view of control, you have input actuators, you got your biological system, all sorts of sensors, and the goal is to figure out networks like that. So if this is a generic bioreactor that can measure everything, these are all the things it can measure. We've measured all of these things, but never at the same time and never in the same system. These are the inputs that you can use as actuators to drive the system. And what you then need is a controller to connect the sensors to the output, and therein lies your model. And you can start getting into deep arguments as to how detailed or physiologically realistic you want that model. There are challenges in control of biology, particularly on the chips I'm talking about. We don't have to go into them in detail. But now let's go to the last part, which is chemostats. So Leo Szilard got a patent on the nuclear chain reaction and couldn't find anybody who wanted to license it. And he then went to Einstein, and it was no Einstein. He says, Al, I need you to take this letter, sign it, and hand it to Roosevelt. So Szilard's letter with Einstein's signature goes to Roosevelt, which launches the Manhattan Project. And Szilard worked on the Manhattan Project, but ended up crosswise with Leslie Groves because Szilard was a kind of a free thinker, probably a bit like Lewis. So Szilard said, fuck it, and went to Chicago and started doing biology and invented the chemostat. All of this is a very sophisticated, no moving parts, low cost, easy to regulate flow regulator. It delivers media at a rate dependent upon the pressure height. This is the chemostat, and it grows yeast. And when the yeast get too populous, they go out into the collecting vessel. So you have a constant volume of continuously perfused yeast. You have commercial chemostats. They were extremely popular in the 60s, but by the 70s and 80s, molecular genetics and well plates took over. But you're basically, all of this stuff is being masked. Heterogeneous batch cultures are masking secondary growth, and the stress effects are hiding differences. So there's a growing recognition that continuous culture is going to be useful. So look at this problem. Here you have a cartoon representation of cells growing in a reactor. They are consuming nutrients and they're producing wastes. And you put in a drug and they consume less nutrients and produce fewer wastes, and you take the drug off. And you have to try to subtract those to figure out what's going on. So in fact, what you really would like to do is look at, I think this curve's upside down.
John, what's the time check? We've got a couple minutes left.
Okay, we're fine. You go to a continuous reactor. The research ones are not that big. They're complicated. They don't scale. And so here's our plan for a 10,000 channel chemostat. If you can build, Ross King wanted me to build him a 4,000 channel chemostat, use mass spec, using solid phase extraction with rapid fire. If you can build one, you can build, if you can build 100 channel, you can build 100 of them. That's the layout for the first prototype. This is the drawings for the first prototype. There are a lot of challenges to make this work. We've come completely now, how do we do biology, down to chemostats. Some of my colleagues say, John, really, you're making things very complicated. And then they ask me to build them a valve. And so I would argue that engineered complexity may lead to simplicity. Those are the lessons we've learned from my group, is that given all of these devices, we can make widgets that work on a lot of applications. These are all the people that have been working with me for the past years. Okay, that's it.
John, thank you so much for the awesome presentation. I mean, it continues to amaze me how you managed to get through so many slides in such a short amount of time. I'm sure you probably have a record on it somewhere. But actually, we've kind of blown through the Q&A part of the talk. We're at 3 o'clock, or we're at 12 o'clock now. So
are there questions?
What we'll do is we'll follow up offline with questions on workplace. And for each of those questions, I can feed them back to John, and maybe he can shoot out the answer for you. I do want to close by just mentioning that we do have this R&D seminar series planned to continue into 2021, and please look out for those invitations and notices. And thank you for everybody that attended.